Yeah, so good afternoon, everybody. If I would like to first summarize like 1 century of brain parcellation basically into one picture. More than 1 century, actually. So as Paula has explained today, it has started with histological work. So the first parceller, we can consider that the first parceller were the first brain cartographer. We wanted to get a map of the brain, like a universal map of the brain. And basically what they did is that they look at brain tissue ex vivo under the microscope, and they look at how cytoarchitecture, for example, change, and they define different brain area based on this change into cytoarchitecture or different feature that they could observe under the microscope. And what you can see here is the drawing from Brodmann, I think. So he basically drew what he could see under the microscope, and then, I mean, you should think of it like it was more than 1 century ago. So the only way to build the map was to draw the map manually. And so this is what they did. And of course, it was quite subjective, and across time, more objective methods have been developed. So now cytoarchitecture work is mainly done by automated approach, in which an algorithm can detect the change in cytoarchitecture, and then the map is built based on a computer approach. So it's not drawn anymore, but the biggest revolution, I think, in the field of brain parcellation has appeared with the development of neuroimaging scanners, because it opened the perspective to acquire a whole brain volume in a few minutes across many different subjects. So it was therefore possible to look at different brain features. And as you may know, brain scanner give us brain images, which are made of voxels or vertex, it might depend. And then we can compare the difference across voxels to define different brain areas based on the different features that we can see in the voxel. And we can build brain map based on those features that we observe in our voxels. And now the richness of MRI scanner is that they offer many different features. So we can look at structural features, functional features, connectivity or more local features, and we can look at differences in voxel across voxels. For all those different features, we can then build a collection of different maps or try to integrate everything into a multimodal map. And this has been the last development, but one of the big advantages of neuroimaging scanners is that now we can look at connectivity. So we can infer connectivity from the measurement that we get out of the scanner. And as Paula said, connectivity is somehow related to cytoarchitecture, and it has been proposed a long time ago as one criterion for defining different brain areas. And the basic idea is that with a neuroimaging scanner, you can measure connectivity. There are many ways of doing so. This is what we're going to go through today. But the basic principle for parcellation is always the same. If you take one region of interest, for example, the hippocampus, you look at the voxels. If you take two different voxels, they might have different profile of interaction with other brain regions. So for example, here one anterior voxel has a different profile of connectivity than the most posterior voxels. If we do that for all the voxels of the hippocampus, we can then label them or cluster them according to their connectivity profile. And then we can distinguish, for example, the anterior from the more posterior part of the hippocampus. Okay, so this is just a basic principle, and this would be the same for any type of connectivity-based parcellation. But there are many different types of connectivity that we can measure from MRI scanners. So there are functional connectivity, which refers to statistical dependencies between brain regions. We can measure them across a range of different tasks. So by looking at coactivation, so this is what Danilo is going to talk about today. But we can also look at functional connectivity as inferred from signal fluctuation at rest. This is what Lucina is going to talk about today. We can also estimate functional connectivity from structural covariance. It has been used in some studies for parcellating the brain, but there are not so much studies. But we could also look at anatomical connectivity directly, structural connectivity, so physical connection between brain region by using diffusion data. And this is what I'm going to talk about today. And the main approach that has been used in the literature based on structural connectivity for parcellate the brain is probabilistic diffusion tractography. And so it has been used for defining different brain areas. And for example, here Thiebaut, Schotten, and collaborators have parcellated the frontal lobe into 12 regions based on connectivity inferred from structural connectivity. So this is really, it opens a lot of application. How does it work? Basically, whatever the type of connectivity-based parcellation approach is always more or less the same procedure. First, you take a region of interest or the whole brain. It depends what you are interested in. Such as here we took the dorsal premotor cortex, so which is an interface between the prefrontal region and the primary motor areas. First, we have to compute the connectivity in the voxel in our region of interest that we call our seed voxels. So our voxel of interest are our seed voxels, and we compute their connectivity with all of the voxels in the brain. And this gives us a connectivity matrix. So for each voxel, our seed voxel within our region of interest, we have a connectivity profile with all other voxels in the brain, or here, all the gray matter voxel in the brain. In other words, we have a connectivity fingerprint for each of our seed voxel in this connectivity matrix. It means that we can use this information, so the connectivity profile of our voxel, to look how similar, dissimilar they are, to compare them between each other, and then to cluster them according to their connectivity profile. So here we look at the similarity, we compute a similarity measure, so for example a correlation, and then we cluster them according to their connectivity profile, and when we remap in the brain, we have different brain area basically. So here, for example, we can subdivide the dorsal premotor cortex into 5 functional subregions. Okay, so that was a basic principle of connectivity-based parcellation. And to do it based on structural connectivity, we can use probabilistic diffusion tractography. So what it gives us is basically tell us how probable is that a connection goes through a given voxels. And there are many different ways of doing it. I would suggest that if you just want to give it a try, if you just want to progressively familiarize with brain parcellation, you want to try, this is your first exercise of brain parcellation, and you want to do it based on structural connectivity, I would suggest that you just use the standard FSL tools because they are well documented. First, what you need is diffusion data, so you can acquire your own diffusion data. But if you do it for the first time and you just want to give it a try, I would suggest that you use open access data. So there are big open access data, such as the NKI sample data or the Human Connectome project, which has very good data, I mean, high quality data. Then you go through the basic pre-processing steps that are well documented on FSL website. Basically, you do the brain extraction with BED, then the eddy current correction, and the registration. This is really standard pre-processing. You can just follow the basic step, the basic parameters, because this is the same, basically, whatever you do. And then you have to run a BED post function, which will fit a probabilistic diffusion model to your diffusion data. And this will give you the input that would be used for the probabilistic tractography of your voxel that you can perform with ProbTrack X2. ProbTrack for probabilistic tractography, X for crossing fiber, and 2 because this is the latest version, I think. And this will give us the probabilistic tractography really of your seed voxel. This is really straightforward. You can follow the parameters that are suggested on the FSL website. The only thing that you need to input a seed mask, and you need to define your target voxels. So the seed mask will tell basically the algorithm where to start a streamline, and the target mask will tell the algorithm where to record connectivity. there are different ways of defining the target mask. Some people choose only white matter voxel, other people choose boundary between white and gray matter voxel. You can try different approach. And what you have to specify as well is the type of output that you want out of this prop track function. And here what we want is a matrix of seed voxel by target voxels. And this is called matrix 2 in FSL. So this basically gives us your connectivity matrix that then you will input into a clustering algorithm. If you look at FSL website, they suggest that if you just want to give it a try to run, for example, a k-mean clustering, there are many different clustering algorithms, but you can, if you just want to give it a try, use k-means. They give even some line of code for doing it in MATLAB, I think. Well, you don't have to use MATLAB, but you can, they suggest it. You just need basically, it's a bit computationally expensive, but you just need to press the button and then you have your clustering. So this is data mining approach. It is, you press the button, it is automated and you get an output. So this is wonderful, it is unsupervised learning, you just put the data in there and you get some output. But I would like to emphasize that it doesn't mean that you have to get rid of your brain, you still need to use it. I would suggest that you still need to supervise the procedure and to think of what you input and what you get out of it, to carefully look at your data and your output. So to give you one example, so for example, here we parcellate the left dorsal premotor cortex, so we use this broad region of interest. Then if you use k-mean, for example, you have to specify the number of cluster that you want. So you can choose 2 cluster, 3 cluster, 4 cluster. It might depend on your a priori hypothesis. And here we ask for 4 cluster. And so we just basically follow the procedure that I just explained, and we run a k-mean algorithm on the connectivity matrix. And this is what we get. So we get 4 cluster, we have 4 cluster, and wonderful, we get this rose trachodal organization for 3 clusters. So we are in the frontal lobe, that's perfectly fit with what is said in the scientific literature. Rose trachodal organization, and in addition to that, we have one new area that we did not expect. So we might find it a pretty name like premotor 66F, and we could write to nature, we have found one new area in the brain. But isn't it a bit suspicious that this area is elongated horizontally along the cortex surface? I think this is where we should be careful, because maybe that might be an artifact. So actually what we did here is that we have just input a big VOI into the procedure and try to get cluster out of that. But one thing that I did not really explain is that when you perform tractography, you need to define your seed voxels, and some people define the seed voxels usually really at the border between gray matter and white matter, because your algorithm has basically to find its way into white matter, so to draw a tract. And the more far away you are from the white matter, the more difficult it would be to draw the path through the white matter. And this is basically what happened here. Tractography failed for all those voxels because we were really far away from white matter. So this is not a new brain area. This is just an artifact of how our parcellation work. Okay, so, well, there are a lot of limitations from the tractography procedure. They might be false positive. There are also a lot of limitations from the clustering or the factorization procedure. They make some assumption on the type of data. So one question that we might ask is, is it at the end really biologically valid what we get out of there, given all those potential artifacts, all potential limitation? And that is actually a very good point. And one way to look at that is what several studies have done is that they have looked at the reproducibility of the pattern that we get out of the parcellation based on structural data, but also based on different type of data. And for example, this is what we did. So we first performed tractography-based parcellation of the right dorsal premotor cortex in 20 healthy young subjects with a spectral clustering, and then later we rerun the same tractography approach in 124 healthy subjects from the NKI data with a K-mean clustering. And the age range was really different. So here it was really only young, healthy adult, and here it was a really broad age range. And we got a similar pattern. So we really get the same type of organization for the two different samples with two different clustering procedure. So it can give us some relative confidence on the biological validity. But one argument also that is actually a good argument is that, well, we still use the same tractography procedure. So if there is a strong artifact, as it can happen in the tractography procedure, whatever the type of sample, whatever the type of clustering approach that we use, it will still be there, and it will even be more likely that we reproduce the same parcellation scheme if the artifact comes from the tractography procedure directly. And for example, looking at stability across subject, this is the same problem. So there are also a lot of studies which try to infer biological validity from the stability across subject with a bootstrapping procedure, for example. But still the question is open that if the artifact comes directly from the approach that we use for computing the feature, it will still be in a different parcellation scheme. So one additional way to try to gain some confidence in the biological validity is also to look at different features. So this is what we did here. So we have parcellate the right dorsal premotor cortex based on structural connectivity, and then we have run a similar approach but based on resting state functional connectivity. And we could replicate basically the pattern that we get. We also did that based on task-based activation data, so meta-analytic connectivity modeling, and we also find a similar organization. So across different different connectivity modality, we always find this organization along an anterior, posterior, and ventrodorsal axis. So it gives us some confidence on the biological validity of connectivity-based parcellation, even though we can never be certain that it is biologically valid what we get out of there. But this is really important question, this biological validity, because there are a lot of perspective for connectivity-based parcellation, and one really exciting perspective is cross-species comparison. So, for example, what Serliani and collaborators have done there is that they have parcellated the frontal lobe of the macaque cortex based on structural connectivity. And they were able to identify different territories in the frontal lobe of the macaque cortex. So this is basically the same approach that we use in humans. So this is based on diffusion data, except that it was ex vivo, I think, but yeah, and a high resolution. And they found this different connectivity-based region in the frontal macaque cortex. So this is pretty nice because it means that this opened the perspective of comparing with brain what we get in humans, and what are the similarities, what are the differences, what does it mean about what makes us human at the end, and how our behavioral functions. Okay, one point that I would like to mention today as well, like Paula has already, I think, risen this point, is the first cartographer have this idea of drawing map of the brain, like well-defined areas, well-defined region, but now we also think of brain organization in terms of gradient. And this is a bit different from the previous tradition, I would say the previous view. So if you look at first parcellation studies, in particular structural parcellation based on structural connectivity. So here, for example, from the Oxford group, what they did is that they were interested into the medial, posterior medial frontal cortex, and they use probabilistic diffusion tractography for parcellating this region. So they just follow the procedure that I explained, and which is actually proposed on FSL website. So they build a connectivity matrix from probabilistic diffusion tractography. Then they build a cross-correlation matrix, so looking at the similarity, which get the similarity between the seed voxels. And then they apply a spectral reordering to reorder the voxel based on their similarity. And what they directly saw here, and that you can now still directly see, is that there are two different groups there. It's quite straightforward. It's quite, and they did it actually visually. They said they look at this matrix and they say, okay, there are two clusters there. This is quite obvious. And indeed, this fit perfectly with the pre-SMA and the SMA. So this is also where we start from, with hard clustering and clear-cut patches in the brain. But now we are progressively moving a bit towards gradients, and this is something that Daniel is going to talk about today, so I won't really go into gradients, but I wanted just to say that it is also possible to use a parcellation approach for studying this gradient in the brain. And this is, for example, what Claude Bajada and collaborators have done. So they were interested into the lateral temporal lobe. They just follow basically the same procedure that I explained. So they compute the connectivity of those seed voxels, the structural connectivity based on probabilistic diffusion tractography with all of the voxels in the brain. And then they have looked at the similarity of their seed voxel. They have run a spectral reordering for reordering the seed voxel based on their similarity. They have assigned a value of those voxels accordingly and then apply a color gradient that fit with those values, and then remap the voxel in the brain. And what you can see here is the gradient that they get when they map back to the brain. So we can nicely see that there is a gradient from anterior to posterior region. So this is one out of the new perspective of brain parcellation, and I would say that it is more a conceptual perspective because it's, I mean, this gives us an idea of how is organized the lateral temporal lobe based on connectivity. But then for using that, for example, if you want to study semantic dementia and change in semantic dementia, this is not very practical, not very useful. So this is really important from a conceptual point of view, but for application, it's a bit more complicated, I would say. Okay, so thank you very much for your attention. I here collected just a few reviews. If you want to get into diffusion MRIs, there is this huge book which has many different chapters that covers. Yeah, Vola, you has read it all? No, like. But it's huge. It's really huge. Like you have everything there, like many chapters covering the many different points. If you want some practical hand-on session, I would suggest the FSL course. Well, I'm not paid by FSL, so I'm not really advertising for FSL, but I know it is well documented, and they have very long experience into diffusion data, and their courses are really nice with really practical session. But there is also other software, for example, MRI Tricks, which is also very well documented. So, and thank you for attention. And if you have questions, please ask.